Greetings, we're back again, part two. Me and Archbishop with this marvelous lesson on immortality. And we know immortality is an indefinite life, past mortal that exceeds mortal life. And as I said, some species have the ability to achieve this by what we call biological immortality.
So great.
Right? We know biology deals with the nature and the study of your cells. All right.
Okay.
The study in the science of the nature of your cells is the only way to see what we call immortality. All right. So all that spook stuff is out the door. You know what I'm saying? They'll tell you right here, they knew about this all the time, that the only way one can achieve, which is for a chosen few will meet to measure up to the bar of achieving this state of immortality through meditation. Meditation is through mind transferring of light through photosphere. It's being able to receive light without actually seeing it through your eyelids. All right. Most of the time we receive our mind transfer information through reading. All right. But we know when we read and focus on what it is that we're reading in lieu of measuring it, bringing clarity to it, it must be concrete, which takes you into dealing with it from a legal, lawful, and historical standpoint. All right, which this is where we come at it with it. This is why we're doing this today, to bring this revelation to you, that let you know that immortality can be achieved through understanding not just through your biological, ancestral or genetic makeup, but also by your not by your genetics or ethnicity, but also by your biological or your cellular lineage. This is what we're trying to wake you up to. Your cellular lineage. All right, let's go to it. So let's go back into biological immortality. Read that from Archbishop.
Wait for it to load up now. All right. Biological immortality, sometimes referred to as bio-indefinite mortality, is a state in which the rate of mortality from senescence is stable or decreasing, thus decoupling it from chronological age. Various unicellular and multicellular species, including some vertebrates, achieve this state either throughout their existence or after living long enough. A biologically immortal living being can still die from means other than senescence, such as through injury, poison, disease, predation, lack of available resources, or changes to environment.
Okay, so we understand that death is non-deniable. Everyone gonna have to make that trip. It's understanding the trip. It's what's beautiful about it. You know what I'm saying? No one can escape this. The understanding to the plane that you're transcending from, it would be stupid to go there without understanding where you're going. Because that's what immortality is based upon. You wish for a future existence without no instruction on where the heck you're going. When you take a trip, you first have a brochure. Yeah, let's go here. Oh, yeah, this looks nice. Versus just going somewhere and don't know where the heck you're running into.
Mm-hmm.
All right? So it says biological immortality is a form of immortality that can be achieved, notwithstanding the injuries that you can occur that can ultimately result in the same destination, such as, like I said, diseases, injury, poisons, predations, lack of available resources, or a change in the environment. All right? And all this denotes to what we call the immortality, going back to your biological understanding, which goes back to what we call cellular lineage or cell lines. All right? Because our existence here on this plane of existence is based off our germ line or our cells. Germ cells or germ line. You have a germ cell or germ line, as well as you have what they call a cell line and a cell on the what you call a cell line. What's the other one? A cell line, yeah, a cell line and a cell lineage. all right, cell line and the cell lineage. All right, it has a descent, ancestry. Let's view that. Right here, biological immortality. We're going to read it right here. Read that for me, Archbishop.
Cell lines. Biologists chose the word immortal to designate cells that are not subject to the Hayflick limit, the point at which cells can no longer divide due to the DNA damage or shortened telomeres. Prior to Leonard Hayflick's theory, Alexis Carrel hypothesized that all normal somatic cells were immortal.
Okay. Somatic cells are not the same as mitochondrial DNA or genomes. All right? Not the same. You must understand that. Your genetic cells or mitochondrial DNA has 23 chromosomes, where somatic cells only have 2. Okay? We'll discuss that in a second. But it says that in biology, cell lines is closely associated with the word immortal, because that's the only way to achieve biological immortality is through your understanding of your cells. Through the understanding of your cells. Let's qualify that by what we call immortalized cell line, through what we call cell lineage. Where it says here, the point at which cells can no longer divide due to DNA damage. Doing DNA damage is what we call radiation. Radiation, damage by radiation is damage to DNA. All right, you must understand that. And DNA, when you see sunlight, visible light, is a result of radiation, ultraviolet rays. That comes from the sun. All right, and we know all life depends upon the rays of the sun. Just like we do, everybody else do. Plants, photosynthesis. All right, let's talk about this. Immortalized cell line. Read that for me, Bishop.
An immortalized cell line is a population of cells from a multicellular organism which would normally not proliferate indefinitely, but due to mutation have evaded normal cellular senescence and instead can keep undergoing division.
Stop. Read that again.
It says an immortalized cell line is a population of cells from a multicellular organism which would normally not proliferate indefinitely, but due to mutation have evaded normal cellular senescence and instead can keep undergoing division.
Okay, cell division. All right. Immortalized cells can continue cell division, i.e. mortifying, what they call Morpheus, changing shape, or what they call mitosis. All right. Immortalized cells are able to continue on to continue to self-divide as a result of what they call mutation. You read it there for yourself. All right. This is because you understand your cell lineage. You understand your cell lineage. What is cell lineage? Cell lineage. Everybody see this is the topic, cell lineage on your left. Read what it says. Early development asymmetries. Symmetries.
In humans, after fertilization, the zygote divides into 2 cells. Somatic mutations that arise directly after the formation of the zygote, as well as later in development, can be used as markers to trace cell lineages throughout the body. Beginning with cleavages of the zygote, lineages were observed to contribute unequally to blood cells. As much as 90% of blood cells were found to be derived from just one of the first 2 blastomeres, In addition, normal development may result in unequal characteristics of symmetrical organs, such as between the left and right frontal and occipital cerebral cortex. It was proposed that the efficiency of DNA repair contributes to lineage imbalance, as additional time spent by a cell on DNA repair may decrease proliferation rate.
Okay, so we know DNA repair in humans after fertilization, the zygote divides into cells. That's called binary fission. That's called binary fission. All right, we'll talk about that in a second. But it says the human after fertilization, the zygote divides into cell lines. As we see, zygote is right here. You see it right here, cell lineage. All right, so let's break this down and understand this once and for all. Once and for all. Let's go there. It said here that somatic cells, where were we at? Biological cells. Oh yeah, biological cells. Cell line. Okay, here we see. I said Violet chose the word immortal to designate cells that are not subject to Hayflick's limit, to point at which cells can no longer divide due to DNA damage or shortened telomeres. That goes back to cell lineage. Cell lineage denotes the developmental history of tissue or organ from the fertilized embryo. This is based on the tracking of an organism's cellular ancestry due to the cell's division and relocation as time progresses. This starts with the originator cell and finishes with the mature cells that can no longer divide. It can no longer divide as a result of what we call DNA damage. DNA damage. DNA damage occurs in cells that originate from what we call mitochondrial DNA, or what we call genomes, because they originate from something else. So let's discuss the origination of where these genomes originated from. But before we do that, let's just discuss those who are subject to that genome, those cells. All right, let's deal with humans first.
Mm-hmm.
We exclude no one.
And brothers and sisters, take this information and you see this is the microcosm, and we represent not only the microcosm, but we make up the macrocosm. We're in unison. You know, everything that's happened in our own reality is a reflection of what's going on within us, i.e. ourselves.
If you look in human, I'm just concerned about the domain. We know we ain't plants, and plants know we ain't humans, and we know we ain't animals. But they all subject to the same domain. They're all subject to the same domain, i.e. that family. As you can see here, the domain under scientific classification is how you pronounce the word?
Eukaryotic.
Eukaryotic?
Yep.
Eukaryotic. The kingdom Animalia. Once again, human beings in scientific classifications fall under the domain of eukaryotic and kingdom Animalia. All right, you see it for yourself. Eukaryote is what's made, is what the cell is made of in lieu of the nucleus. The nucleus of your cell contains eukaryotes. all right, which gives the ability for the cell to divide, i.e., as we said, when the cell can no longer divide, it's a result of cell breakdown. But when it's able to divide, that's the result of what we call eukaryotes. All right, we'll discuss that in a second. We've seen human, let's look at animals. Go right to the box. Scientific classification, domain, eukaryota. Everybody see it for yourself. Domain, eukaryota. That's animals. Let's do plants. Can you see that, Archbishop?
Yes, I can see it.
Okay. Plants, read that for him.
Plants are eukaryotes that form the kingdom of plantae. They are predominantly photosynthetic. This means that they obtain their energy from sunlight using chloroplasts derived from endosymbiosis with cyanobacteria to produce sugars from carbon dioxide and water, using the green pigment chlorophyll. Exceptions are parasitic plants that do not contain chlorophyll and obtain their energy from other plants. Historically, as in Aristotle's biology, the plant kingdom encompassed all living things that were not animals, including algae and fungi.
Archbishop, you can hear me?
Yes, I can hear you.
That's fine. So we know that animals, plants, and humans fall in the domain under the scientific classification as eukaryotes. Isn't that right? Did they say that?
Yep.
All right. Anybody dispute that? Please do. All right. But we just showed you. Humans, animals, plants fall under the domain of the set of scientific classification as eukaryotes. So let's find out what that is. Since we're trying to make this shift to immortality on a biological level, all right, on a biological level, because we understand that somatic cells are considered what they call immortal, whereas though genomes are not immortal. And all cells, whether male or female, are genomes. Why? Because they are eukaryotes. Let's prove it. Let's look at karyotypes, first of all. What is karyotypes? Read that first paragraph, Archbishop.
Karyogram or ideogram is a graphical depiction of a karyotype. A karyotype is the general appearance of the complete set of chromosomes in the cells of a species or an individual organism, mainly including their sizes, numbers, and shapes. Karyotyping is the process by which a karyotype is discerned by determining the chromosome complement of an individual, including the number of chromosomes and any abnormalities.
Okay, that's called mitosis, morphing, or what they call mutation.
Mm-hmm.
Mitosis, morphosizing, or mutation. All mean the same. All right. So karyotypes is general appearance of the complete set of chromosomes in cells of a species. When we say species, we're talking about all those that's subject to the domain of eukaryocytes. All right, because these are the karyotypes. Now let's look at the eukaryocytes, which humans, animals, and plants fall into. If you want to be immortal, you better follow the instructions on how to get there. Read that.
The eukaryotes constitute the domain of eukarya, organisms whose cells have a nucleus. All animals, plants, fungi, and many unicellular organisms are eukaryotes. They constitute a major group of life forms alongside the two groups of prokaryotes, the bacteria and the archaea. Eukaryotes represent a small minority of the number of organisms, but due to their generally much larger size, their collective global biomass is much larger than their global biomass is much larger than that of prokaryotes.
Okay, so eukaryotes are organisms whose cells have a nucleus versus prokaryotes, which don't have a nucleus. That's very important to understand the two. Very important to understand the two. All right, why do I say it's very important? Let's find out. Let's just find it. I don't want to miss nothing. Okay, let's move there. Let's go here. Somatic. Read that for Marcus.
Somatics is a field within bodywork and movement studies, which emphasizes internal physical perception and experience. The term is used in movement therapy to signify approaches based on the soma, or the body as perceived from within.
Okay, stop. For the body as perceived from within. Very, very important. That's what somatic is. Now let's look at genome. Read that. You can see that?
Yep. The fields of molecular biology, in the fields of molecular biology and genetics, a genome is all the genetic information of an organism. It consists of nucleotide sequences of DNA or RNA and RNA viruses. The nuclear genome includes protein-coding genes and non-coding genes. Other functional regions of the genome, such as regulatory sequences, see non-coding DNA, and often substantial fraction of junk DNA with no evident function. Almost all eukaryotes have mitochondria in a small mitochondrial genome. Algae and plants also contain chloroplasts with a chloroplast genome.
Okay, so you got karyocytes and then you have prokaryocytes. What are prokaryocytes? Because eukaryocytes have a nucleus. All right? And the nucleus contains all the genetic information of a cell from the point to its cell division, till it can no longer divide. When we know it can no longer divide, that means there's a breakdown in what they call the cellular structure as a result of what we call DNA damage. Result of DNA damage. But it says that somatics have the ability to mutate, morphosize, or mitosis as a result of their genetic makeup. All right, that's what we're going to get into. All right, that's what we get into. That brings us here to prokaryocytes. What are prokaryocytes?
A prokaryote is a single-cell organism that lacks a nucleus and other membrane-bound organelles. The word prokaryote comes from the ancient Greek pro, which means before, nut, kernel, and the two empire system arising from the work of Edward Chatton. Prokaryotes were classified within the empire prokaryota, but in the three domain system, based upon molecular analysis, prokaryotes are divided into two domains: bacteria, formerly eubacteria, and archaea, formerly archaea bacteria. Organisms with nuclei are placed in the third domain, eukaryota. Prokaryotes evolved before eukaryotes.
Okay, so that's getting to what we call cell line or cell lineage or germ line or germ cells. There's distinction between cell lines or cell lineage and germ line or germ cells. So let's expose both of them. All right, let's expose, because we know that eukaryotes is where the cells start to split and to divide. All right, and we know that's a result of, as they showed you in the picture, between what we call the germ cells and sperm cells and what we call ovums are germ cells from the germ line. Let's prove it. Because these are what you call genomes, mitochondrial DNA. One coming to short on this one. Read that first paragraph.
In cellular, eukaryotes are made of two fundamental cell germ types, germ and somatic. Germ cells produce gametes and are the only cells that can undergo meiosis as well as mitosis. Somatic cells are the other cells that form the building blocks of the body, and they only divide by mitosis. The lineage of germ cells is called the germline. Germ cell specification.
Read that sentence again. Lineage.
Say it again.
Read that part again. The lineage of a germ cell.
Oh, the lineage of germ cells is called the germline. Mm-hmm. Germ cell specification begins during cleavage in many animals or in the epiblast during gastrulation in birds and mammals. After transport involving passive movements and active migration, germ cells arrive at the developing ganodes. In humans, sexual differentiation starts approximately 6 weeks after conception. The end products of the germ cell cycle are the egg or the sperm. Under special conditions, in vitro germ cells can acquire properties.
That's fine. That's fine. Let's go here now. So we understand that genes deals with the mitochondrial DNA that we see on this and the science that we study and dealing with biology. Genes are based off the instructions that were given inside the nucleus of a cell, all right, which are called eukaryotes. But you have prokaryotes that don't have a nucleus. That's very important because the prokaryotes is what they call nuclear DNA. And nuclear DNA is a result of what we call nuclear fission, which turns into what we call binary fission, which results in your cells being able to multiply, which we'll validate and qualify right now. All right. There we go. Nuclear matrix.
That's it.
Go ahead. Read this.
In biology, the nuclear matrix is a network of fibers found throughout the inside of a cell nucleus. after a specific method of chemical extraction.
Stop. In biology, the nuclear matrix is the network of fibers found throughout the inside of a cell's nucleus after a specific method of chemical extraction. So we know a cell's nucleus automatically denotes to eukaryotes, what they call eukaryotes. How you pronounce the word again? Eukaryotes. Because eukaryotes have a nucleus. Prokaryotes or karyocytes have a nucleus. Prokaryocytes don't have a nucleus. All right. So that means that between the prokaryocytes and the eukaryocytes, it created what they call a nuclear matrix. Does that make sense? Is there, Archbishop? It said in biology, the nuclear matrix is the network of fibers found throughout the inside of the cell nucleus after a specific method of chemical extraction. According to some, it is somewhat analogous to a cell cytoskeleton. So it represents the framework. It represents the framework because the biology of the nucleus is part of what they call a nuclear matrix. And we know a nuclear matrix is based off nuclear DNA. You have binary DNA, which is called genomes, right? That's our DNA, but that DNA received instructions from what we call nuclear DNA, and that resulted in what we call nuclear fission. And then those instructions were enveloped in what they call nuclear envelopment to the cell through its nucleus. You don't believe me? Let's qualify it. How much time we got?
5 minutes.
Okay. Let's see. Right here. Nuclear, let's start here. If you can remember in the video, and when I showed you when we first started, it shows you the origin of the cells, which that originated from your mother and father, and it showed you an image look just like this. Showed you an image look just like this. When you watch this video over again, please look over it. Read the nuclear envelope and what that says.
The nuclear envelope, also known as the nuclear membrane, is made up of 2 lipid bilayer membranes that in eukaryotic cells surround the nucleus, which encloses the genetic material.
Read that again.
It says the nuclear envelope, also known as the nuclear membrane, is made up of 2 lipid bilayer membranes that in eukaryotic cells surround the nucleus, which encloses the genetic material.
Okay, we know the genetic material is what they call genomes or DNA. We know that the genetics, this is qualified, genetic material in the field of molecular biology. So I know what I'm talking about. In the field of molecular biology and genetics, a genome is all the genetic information of an organism. It consists of nucleotide sequences of DNA. The nuclear, you hear that word, nuclear. Genomes include protein coding genes and non-coding genes, other functional regions of the genome, such as regulatory sequence, and often whatever else they said. All right. So it says nuclear envelope, also known as nuclear membrane, is made up of 2 lipid bilateral membranes that the eukaryotic cells surrounding the nucleus. And we know eukaryotic deals with cells that have a nucleus. And within that cell, through the nuclear envelope, contains all the genetic material, which you call genome, i.e. your DNA. For all those that exist as in the domain of, on the scientific classification of what they call karyocytes. All right, that includes animals, plants, and humans. Animal, plants, and humans. But that's based off the genetic material here that was created through the nuclear envelope of the information, i.e., the light information that gives that entity or that manifestation on this plane to be able to exist, such as humans, plants, and animals. It's the genetic information for them to exist. All right. So we're dealing with R's, and we're going to R's in part 3. All right. So we'll see you in a second. Peace.